Hi, my name is Taryn Connolly, and for my protein project, I'm working with Piezo 1. Piezo 1 is located in the cell membrane of red blood cells, skin cells, and smooth muscle cells, who all require the protein for detection of mechanical forces in the environment. These mechanical forces then trigger signaling, which allows the flow of ions across a membrane. A mutation in this protein is most commonly associated with exerocytosis. It is a rare condition characterized by dehydration of red blood cells due to a defect of increased permeability, which results in a loss of potassium and a gain of sodium. Before the mutation occurs, the sequence is basic and polar charge. These characteristics remain the same even after the mutation occurs. Even though these characteristics remain the same, the change in the sequencing is still large enough to cause damage to the organism. The overall shape of this protein is a 3D blade or fan-like structure, with 3 different subunits characterized by my 3 different colors here. The 3 subunits are all joined together at the middle of the structure, which creates the ion channel, and then the remaining part of the subunit curves into a blade shaped. The amino acid sequence for the primary structure is represented by the pipe cleaners. And the secondary, tertiary, and quaternary structures are represented by the twisting subunit ends. A mutation in the Piezo 1 gene affects the protein structure by altering amino acids. I have represented the mutation in the Piezo 1 gene with these blue beads seen here on the pipe cleaners. The mutation of amino acids within this gene is a gain-of-function mutation which affects the protein function by adjusting the frequency and consistency at which the central ion channel opens and closes. The inconsistent timing and continuous opening of the channel now shows what leads to a fluctuation of decreased potassium and an increase of sodium. When the red blood cells experience a loss of potassium, they also experience a loss of water, which causes them to become severely dehydrated, and then when the cells experience dehydration, they shrink and undergo hemolytic anemia, meaning that the cells will be destroyed faster than they can be produced, resulting in a low red blood cell count. This affects the organism as a whole, as the body will struggle to carry oxygen efficiently due to the decreased red blood cell count, which will inflict physical symptoms on the body. The mutated amino acid is located around the pore region domain, as seen here by the blue beads. Now, obviously, within my model, there are going to be some limitations, as it is not an actual protein. So therefore, it is unable to perform the actual physical responses you would typically see from the mutated protein. And of course, it is not on scale either with the size of the protein that exists in our bodies. Additionally, another limitation on this model is that it is unable to be altered back and forth efficiently enough to demonstrate all primary, secondary, tertiary, and quaternary structures. Thank you for your time today.